Good afternoon and a very warm welcome to all. Today we are going to discuss about Anoxomat, and it comes under the anaerobic culture methods, which include the combustion or removal of oxygen with other gases. And in this Anoxomat, it is a jar system that offers an alternative method utilizing evacuation replacement method developed by McIntosh and Fields. Yesterday we have discussed about McIntosh Field jar and candle jars. Today we are discussing Anoxomat. It can create standard or user-defined receipt atmosphere, and it produces microaerophilic and anaerobic organisms within the same system. The advanced Anoxomat, it is an automatic microprocessor control system for the cultivation of anaerobic, microaerophilic, hypoxic and capnophilic bacteria. And this Anoxomat's evacuation replacement methodology rapidly removes an oxygenated environment from the jar and replaces it with a precise amount of an anaerobic gas mixture. After this anaerobic cycle takes place, a mere 0.16% residual oxygen content is left within the jar, and this is also removed by a small palladium catalyst. By controlling this procedure with a microprocessor, so in advanced Anoxomat, a microprocessor is present which controls the procedure, the user can define exact atmosphere required in the jar, while other utilizing the minimum amount of jar to accomplish this. Saving, and what are the advantages? It saves the laboratory time, effort and money. It can create standard or user-defined receipt atmospheres, thus provide either microaerophilic or anaerobic organisms within the same system. Complete anaerobiosis can be reached within 3 minutes. It is faster than other standard methods for unmatched organisms' quality and recovery. The operator can count on the results, since the Anoxomat's quality assurance system ensures that jar atmosphere is precisely as required and warns of any types of leakage. The Anoxomat system small footprint reduces operating space by 60%, and its ingenious technology saves laboratories 70% of money wasted on consumables. And the Anoxomat can automatically record user and specimen information, date, time and ending gas mixture in the jar. And thus it eliminates the transcription errors within the optional use of barcode scanner or printer. So these are all coming under the removal of oxygen gas with and filling it with other gases. Second method is chemical methods. The method is employed in different modification that can provide anaerobiosis. The anaerobic conditions are obtained by the absorption of the gases by alkaline pyrrole solution and also in the moat surrounding the cell of the water. The amount of pyrogalol used presents an excess over that required to absorb the small quantity of oxygen present in the part of the chamber not occupied by the pyrogalate and water. The total volume of chamber, it is approximately 5 cc, but the prepared chamber contains less than 2 cc free air space. These relations ensure low oxygen concentration in the prepared chamber during incubation. The chamber moat presents approximately 3 square centimeter surface area for the oxygen absorption by the pyrogalate. The large surface exposure gives a rapid absorption of oxygen, observation of the culture, and it is made by means of light passing through the inner cell of water, as in the case of usual chemical chamber preparation. Loosening of the inner ring, which may result in the entrance of the dark pyrogallate solution into the inner cell, will interfere with the illumination subject. Observation of the culture is made by means of light passing through the inner cell of the water, as in the case of usual moist chamber preparation. The loosening of the inner ring, which may result in the entrance of the dark pyrogallate solution into the inner cell wall, it interfere with the illumination of the object, and this will occur in poorly prepared chambers. That is a disadvantage. A small amount of carbon monoxide is also formed during the reaction, and it may be inhibitory to some bacteria. And the evaporation and change in the culture medium is reduced to a minimum by saturating the chamber with water vapor from the inner cell, which presents a large surface of water. The anaerobic chamber was used successfully to bring the growth and development of obligate anaerobes, namely Bacillus tetani, and not Bacillus tetani, sorry, Clostridium tetani and Clostridium botulinus. There is some mistake in this. So obligate anaerobes, namely Clostridium tetani and Clostridium botulinum. The photomicrograph of the culture could be made directly at different intervals. There is a mistake, okay? And this is the chemical method using alkaline pyrogallol. This is the photo. This is the dish. Next is about spray anaerobic dish. These are older methods. That's why the clear pictures are not available. It is a glass dish with bottom partition into two halves. Top portion is accommodating half of the petri dish carrying the medium. The pyrogallic acid and sodium hydroxide are placed in the separate halves of at the bottom of the dish. The inoculated plate is inverted on the top of the dish and is sealed completely. The dish is then rocked to mix the reagents, thus producing anaerobiosis. And this method is not in use now. Next is a simple modification. A simple modification, it consists of a petri dish between the two halves of which it is inserted a metal disc of slightly large diameter with a hole in the center. And this metal disc, it is attached to the bottom half with plasticine. So plasticine is used. Through the central hole, a few pellets of sodium hydroxide and 10 ml of 10% solution of pyrogallic acid are added. And the inoculated half of the petri dish is then inverted on the metal disc and it is sealed tightly. So another method employs a disc of filter paper having the same diameter as that of the petri dish. And this is placed on the top of one half of the dish, and a mixture of pyrogallol and sodium carbonate in dry powder form is spread on it. The inoculated plate is inverted over the filter paper and is sealed tight with molten wax. The pyrogallol mixture, it is then activated by moisture within the closed system, and a complete anaerobiosis can be achieved within 2 hours. Next is the modification of this method, that is Rosenthal's method, that is alkaline. Instead of alkaline pyrogallol addition, anaerobiosis has been produced within the jars with a mixture of chromium and sulfuric acid or lamb, or with yellow phosphorus. So this method is Rosenthal's method. This can be used instead of yellow phosphorus. So this method is a mixture of chromium and sulfuric acid or with yellow phosphorus, and this method is called Rosenthal's method, and this method can be used instead of alkaline pyrogallol. So today's class is up to this. In the next day we can discuss about gas pack, biological methods, and the anaerobic culture methods using reducing agents, and also certain two types of medium which are involved in the anaerobic cultivation. Thank you.